I will try to refresh your memories from the specificity of Proto-Spacer. Okay? So I hope you remember the Proto-Spacer. It is the spacer or Proto-Spacer, the same thing. Okay? And that is part of gRNA. And that part comes as a target sequence. And it is usually 20 nucleotides long that we have designed on the target DNA, where we want to do some modifications. Okay? And if you remember this is again the slide which shows nucleotide preferences at very specific positions. Okay? And if we look at the PAM site is at 3' end on the target DNA, not on the gRNA. Okay? The PAM site, we look for it on the target DNA, and then we take 20 nucleotides towards the 5' end of the PAM site as spacer or Proto-Spacer. Okay? 12 nucleotides towards the PAM site, they represent the seed region. It means they are the most important nucleotides for the Proto-Spacer specificity. And the 8 nucleotides remaining, they are known as non-seed region. Okay? And some variability in that sequence may be accepted by the gRNA. But this variability always results in non-specific binding. On one side, this is a take-home message. On the other hand side, there are preferences of nucleotides at certain position for the specificity of gRNA as well. So at position number 20, so they counted from 5' end site, and towards the PAM site, it is the maximum number. So towards the PAM site at position number 20, the highest specificity is obtained if the first 20th nucleotide is either an A or a G. Okay? This is rejected, the specificity is rejected if there is a T or a C. C completely disfavors, okay, the specificity of gRNA Proto-Spacer for binding to the target site. So at position number 20, we always keep, okay, either an A or a G. Similarly, at position number 16, we can see that there is a variation, like a C is preferred and a G is disfavored. Okay. Similarly, a C at position 17 is more preferable and a G at position 17 is disfavorable. Some other cases are also there. In case of nucleotide number 14 of the Proto-Spacer, it should not be G. It should be preferably A. Okay. So this is what you consider when you select a Proto-Spacer. Okay. And how you select it, we will talk about that later on. But this specificity tells us about certain rules. The rule, like the 20th nucleotide must be either a G or an A. So we make it a rule. Okay. Similarly, we say non-seed region, we can accept some nucleotides there which may be disfavored or may be favored, but does not matter. But on this side, they are most important. Okay, and this is the seed region, and the seed region must have the most favored nucleotide for the gRNA. Okay, so this is what we talked last time, and then we talked about the 4 different rules. The rule number 1 was the 3' end of the DNA target sequence must have a protospacer adjusted motif, PAM. Okay, so the target DNA must have a PAM at the 3' end of the protospacer. The 20 nucleotide upstream of the PAM sequence would be the target sequence that Cas9 nucleus will cleave approximately 3 bases upstream of the PAM. Okay, so if this is the PAM site, the Cas9 will cut 1, 2, and 3, between 3 and 4. Here it will cut. Okay, so this is another inference that we already have. Okay, so what we do is that this is our target sequence, for example, and then we look for NGG. If we just look for GG, we will find on a very small sequence, the G is repeated 3 times. So we have 3 PAM sites here. We can design our protospacer, okay, upstream towards the 5' end. We can pick the 20 nucleotides here, or we can pick the 20 nucleotides here, or we can pick a 20 nucleotides here. And then we analyze them, all of them. Which one of them meets the specificity issue at nucleotide number 20? It should be an A or a G. So if we take this one as a protospacer, we must have an A or G at this position. What about this case? There is no A, no G. So we will reject this protospacer. Here, we can take this as a protospacer because it has got a G here. We need either an A at 20th position or a G at 20th position. Similarly, at position number 17 and 16, we can look for the other favorable nucleotides and manually we can select that. We must have the knowledge for selecting these protospacers manually. We can do it through software as well. They can also make suggestions, but still you have to look at that which one is the best protospacer for that. Okay, so you must have the capability to manually select the protospacer, which is always good. Okay, so that you can yourself analyze the protospacer proposed by the software. Okay. The second rule was the PAM sequence itself is absolutely required for cleavage, but it is never a part of gRNA. Okay. So what we include in the gRNA towards the CRISPR region is this protospacer, which is 20 nucleotides upstream to the PAM site. We don't include PAM site inside this protospacer. Okay, we just take the nucleotides, 20 nucleotides upstream to the PAM site. This was the second rule that we learned. The other thing was, what we learned was that this gRNA expression is controlled most oftenly by 2 different promoters. And these are either U3 promoter or U6 promoter. The rule for a U3 promoter is that there should be an A. And here you see the reversal. In the previous slide, this was position 20, okay? And this was position 1. So you can take this position as 1 or 20. At position number 1, which is close to the MGG site. That is the target site. So just adjacent to the PAM sequence, the first nucleotide must be an A or a G. A in case of U3 promoter, G in case of U6 promoter. Okay, so the first nucleotide close to the PAM site should be either A or G, and you can take it as a position 1 or position 20, whatever you want to remember. Okay, but the inference is the first nucleotide adjacent to the PAM site should be either an A or a G. A if you are using U3 promoter, and G if you are using U6 promoter. Okay, and now position 17, if you take the first nucleotide as 1, position 17 must have again an A, okay, in case of U3 promoter, and a T in case of U6 promoter. Is it clear? So a gRNA, what is a gRNA? gRNA is protospacer plus CRISPR RNA plus tracer RNA. Okay, so 3 different components represent a complete gRNA, and the gRNA is expressed under a specific promoter. You have a promoter, and then you take these 20 nucleotides as the target, and gRNA scaffold on the upper hand side towards the target, it has the CRISPR RNA, and below that it has a tracer RNA. Okay, and then a stop codon. So that this is the RNA which is produced, and it makes a total gRNA which will be functional for targeting a specific gene. The other thing was that multiple gRNAs per target gene, typically ranging from 3 to 8, should be employed. Okay, it means that, in other words, if you have a target DNA, which is say 100 nucleotides, 200 nucleotides, 500 nucleotides, instead of selecting a single protospacer, you must design at least 6, 7, 8, or 10 protospacers. And you should try each one of them, because you don't know which one of them is going to work the best. So it's a random game. You need to design more sequences as a protospacer, okay, and try each one of them and see which one is highly specific. And this can actually be determined by some of the softwares that I will tell you later on. But first of all, you must know that you need to design more than one protospacer, and I would prefer that after identifying the PAM site, select manually more number of protospacers close to the region that you want to modify. Okay, whether you want to insert a gene there or you want to repair the DNA at that position. So you are not exactly selecting the target site. You are always either on the 5' end, I mean you are away on the left, or you are away on the right from the target sequence. Okay, so you select it away, and we will practice it a little bit later on. Okay, so these were the 4 things that I told you last time, and I hope now you have recalled them. And now I'm coming to the enzyme part. There were several parts. In the beginning, I said I will split the lecture into several regions, the basic components. And then we have talked about the gRNA. Now we are going to talk about Cas9. Okay, what is this enzyme? Okay, This enzyme is produced actually by several different microorganisms. Staphylococcus sp, Streptococcus sp. So these are two different organisms. Both of them, they produce Cas9. Okay. Neisseria, meningitis, NM. Treponema, denticola, TB Cas9. Cas9 is from many different organisms. The most common Cas9 that we talk about is sp Cas9. If you look at the PAM site of this Cas9, it is NGG. This is what we most oftenly talk about, and you must know that it is from Streptococcus pyogenes, and this is from Streptococcus thermophilus. Now, what's the difference between why should we use Cas9 from Streptococcus pyogenes? Look at here. NGG. How many are the specific sequences? Many specific sequences. Here there are two. N is any one of the four, and there are two nucleotides which should be very specific. Okay, and then in this case, there are four nucleotides which are specific and two are non-specific. In this case, again, four nucleotides are specific and four are non-specific. One non-specific and five specific nucleotides. So, which one of these sequences will occur more frequently on the genome? Can you predict it? Can you guess it? Here there are two, here there are four, and then five. Which one of these occur more frequently on any chromosomal DNA?
sp Cas9.
GG, two nucleotides. There are more chances that two nucleotides can occur repeatedly on a genomic DNA. If you talk about four, the occurrence of the four nucleotide repeats, they will be far apart. Okay, so there are lesser number of chances that you see a PAM site of ST-Cas9. And there are far less chances of finding a TD-Cas9 PAM site because it requires a much longer specific sequence for PAM. That's the reason SP-Cas9, the GG, more probability, high probability you will find. And if you remember, in one of the slides, we have a short sequence and we have three different pairs of Gs at different positions. More frequent are the palm sites, better it is. You have more options to make proto-species frequently, okay, close to the desired position where you want, okay. So that's the reason this Streptococcus pyogenes Cas9 has become very popular, okay? Because of the shorter PAM site sequence and greater of frequency of occurrence of these nucleotides. Okay, so now you remember that Cas9 does not mean it's coming from just, it's a single enzyme and it's identical for every organism. It's different. It's different from different microorganisms. And because it is different from different microorganisms, it has different band site as well. So you cannot use a SP-Cas9 for this band site. Okay? SP-Cas9 will always nice and GG. And always look at the Cas9 that you are using in your experiment. Is it SP or something else? If you are using SP-Cas9, then you should look at the PAM sites which have a sequence of NNAGAA. Okay, and then design a protospacer of 20 nucleotides upstream to that sequence towards the 5' end. Okay, so always look what Cas9 have your laboratory got. Is it SP or SP? Okay, these are mostly available around in the lab. And accordingly select the PAM site for making a protospace. So this is the most important part about the Cas9 protein. Okay, I'm not going into the sequence and the tertiary structure of this protein, but the most important thing to remember in case of CRISPR-Cas system is that Cas9 is class 2 DNA genome editing system. It's 3 component, but Briner made it 2 component, and it generated an ease for us that just we need to design a protospacer, put it upstream to the pre-designed CRISPR and tracer region, clone it under a promoter, and that's all. Our gRNA will be ready from there. Now, the biggest problem with CRISPR-Cas9, just like the zinc finger proteins, is its non-specific binding. Non-specific binding means genomic DNA is a big molecule. Billions of nucleotides there. 20 nucleotides, there are chances that the sequence is very similar to the selected protospacer can also exist at some other chromosomes, at some other places. And the protospacer that you design can also bind with those sequences which are similar to your protospacer. So there are a lot of chances. Chances in dozens, chances in hundreds, and chances in thousands. It depends from one protospacer to another protospacer. Okay? The question is, can you have a longer protospacer? The answer is no. Okay? The protospacer must be 20 nucleotides long in case of CRISPR-Cas system, when we are using Cas9. So, people, to increase the specificity, what does non-specific binding of the protospacer mean? The non-specific binding of the protospacer will always result into non-specific cuts by the Cas9. So if the genomic DNA is cut at the required site and we want to manipulate something there, it's okay. This is what we want. But if the genomic DNA also gets cut at dozens of other sites simultaneously along with the specific site, so we can manipulate the specific site, but at the same time, we are disturbing or deteriorating the genes where, or the other DNA where the other non-specific cuts have been placed by Cas9. And that is a disaster. That's a big problem. And to address this problem of specificity, the scientists, they have worked through different means. Okay? So, and they have produced modified CRISPR systems And I'm going to tell you what are those modifications they have done. The first modification that you know is it is from a 3-component system to a 2-component system just by adding a loop sequence between the CRISPR and tracer molecules to join them together. Okay, so now the CRISPR-Cas system is a 2-component system that we use in the laboratory. Other modifications have been done to the CRISPR-Cas system that we are going to study. The first one is Cas9n, or it is also known as paired Cas9 nicases, that you don't use a single gRNA. You use a pair of gRNA. And a gRNA is always accompanied by a Cas9 protein. The gRNA alone cannot do anything. gRNA just takes the protospacer to the target site, nothing else. And Cas9 is the protein that binds to the whole complex of gRNA and target DNA and makes a cut. Okay. So what they did was that, you know that Cas9 cuts 3 nucleotides away from PAM site, right? On both the DNA strands. So what they did, they mutated the Cas9 system. And in that mutation, what did they do? They modified the Cas9 by substituting the aspartic acid, amino acid, at position number 10 to alanine. Okay, that is a D10A mutation of Cas9. So what happened by this mutation? Cas9 started cutting a single strand instead of cutting on both sides of the double-stranded DNA. Okay, so if you have this Cas9n expressed in a genome, which is also expressing a gRNA, so the complex will go to the target site and will make a single-stranded DNA cut, not a double-stranded. So the double-stranded DNA will not break and open into 2 pieces. The nick will be produced. So I said, this modified CRISPR system, it utilizes a pair of gRNA, okay, along with a mutated Cas9. This is one gRNA molecule, the other gRNA. They are in close vicinity. So you design the 2 protospacers in close vicinity, okay? And you use them along with a Cas9n enzyme. Cas9n means it's a mutated, and it will make a single cut in the double-stranded DNA. So you get a single-stranded cut here, and you get a single-stranded cut here. So there are very few chances that the pair of the gRNA can bind non-specifically at other places. Okay. Only the DNA will have a double-stranded break when gRNA1 binds in close vicinity to gRNA2. Okay. If this gRNA1 binds at 10 or 20 or 100 other places on the genome, it will make a single-stranded cut. The DNA, double-stranded DNA, will not fall apart. Okay, so there is no damage to the double-stranded DNA. There will be no mutations at that non-specific binding site. Only if the 2 gRNAs bind close to each other and make a single-strand cut, you can see the cohesive ends will be generated. When you have cohesive ends which are complementary to each other, it makes homologous recombination the method of choice. Okay, it facilitates homologous recombination. If you generate blunt ends or the ends which are not compatible, then you introduce mutations in the genome, which is not desirable. Okay, so this first modified system is really very nice, very nice. It reduces the non-specific cutting of DNA by 1500-fold. Okay, it means your target is now highly specific. You will have very few cutting sites around the DNA at other positions, and you are hardly introducing any mutations in the target genome. Is the mechanism clear to you?
Yes,
Hello?
sir.
Okay. Okay, so this is one of the modifications that scientists have made. The other modification, different laboratories, they do different things. Okay. So what they have done, in this modification, what was lost by Cas9? Cas9 lost its capability to make a cut in the second strand. Okay, so they modified the Cas9 and further, and they took away its capability to cut the double or single stranded DNA at all. And they generated this mutant known as dCas9. What dCas9 is capable of? It can bind to the DNA, it can bind to a gRNA complex, but it does not cut the DNA at all. Okay. Now you know that the Cas9 and the gRNA complex, they actually are sequence specific. The proto-spacer will bind to the target sequence, so it's sequence specific binding. And non-specific binding will always be there, no doubt. They took one thing from the previous modification, that they made two pairs of the gRNA just like the earlier modification. So they have a pair of gRNA designed at close vicinity of a sequence that you want to modify. Okay, one at the left side of that sequence, one to the right sequence side. But Cas9 is not making any cut. What they do? They developed a fused Cas9 protein, a fusion protein. So what they had, a mutated Cas9 that had lost the capability to cut the DNA, and then they joined it with half of the protein of fork-1 nuclease. It's a kind of restriction enzyme. Restriction enzymes, they cut the DNA. So fork-1 is one of the restriction enzymes. They took half of the protein, half of the amino acids on one gRNA, one Cas9 protein, and half of the RNA, of the DNA. It's actually, this enzyme works not as a monomer, but this enzyme works as a dimer. Okay, so one dimer is fused to Cas9 here. The same dimer is fused to the other Cas9 molecule. It means both the dimers, they are identical. Monomers, they are identical. But when the two monomers, they come together, they make a complete nucleus. And this nucleus is capable of cutting the DNA, the dimeric form, not the monomeric form. So the Cas9 joined to the monomer of fork1 will not cut the DNA until an other set of Cas9 and gRNA having a monomer of fork1 comes close, joined there, and the two monomers, they are aligned together to make a dimer. And when the dimer is formed, it will cut the DNA at that particular position. So again, the specificity issue has been addressed by taking a pair of gRNA, so that the chances of non-specific cutting could be reduced, just like in case of Cas9 and the Cas9. It has no capability to cut, so they have taken help of fork1. So a modification is a little advanced from there. Cas9, absolutely no cut in this case. Fork1 has been utilized. Restriction enzymes, they generate sticky ends. Okay. Similarly, the fork1 generates sticky ends, and those could be used for cloning a DNA fragment having fork1 sticky ends at the terminal ends. And that is actually a homology-based recombination, HR. So this also supports homology recombination. It means that in the DNA, the pair has been actually the major concern of CRISPR-Cas9. But later on, it was used for cloning of the foreign acids based on the feasibility that DNA can be cut to generate cohesive ends. And you can insert a DNA fragment with the same cohesive ends, and that can get integrated here. So that's a transgenic technology, which was actually taken up through this. So what is the case here? A catalytically inactive Cas9, that is known as Cas9, is fused to half of the fork1 nucleus, means one of the monomer of fork1 nucleus inside. Okay? So Cas is a defective Cas9. Defective Cas9 means it has no potential to cut the DNA, but fork1 nucleus monomer has been fused with it, so that when it makes a pair with the other gRNA having the other half of the fork1, they can make a double-stranded DNA cut. Okay? So when fork1 dimerizes, it can generate double-stranded break at specific sequence, because fork1 recognizes a very specific sequence. And now you must consider that the site that you want to modify must have a fork1 site and a PAM site as well. Okay? So this system is actually not that popular as is the previous system, because it requires only the PAM sites, and one cut by one Cas9, the other cut by other Cas9. So this system can be used only occasionally when you have both the fork1 site along with the PAM1 site in close vicinity to your target sequence. Okay, remember that. This is not a modification, but this is another CRISPR-Cas protein-like protein, which is known as CPF. Okay, CPF1 is actually coded by another organism whose name is actually below the panel. Okay, so you can see it later on when you will be watching the video. Okay, you can go for that. What it means is this is an other kind of Cas9. It's entirely a different name. Class 2. Okay, this is class 2 system. But Cas9 is a different protein. CPF1 is a different protein. But both of them, either Cas9 or CPF1, they recognize a PAM site and they cut the DNA double-stranded DNA when it is a native enzyme, whether Cas9 or CPF1. What the difference between Cas9 and CPF1? You should be able to answer it. Cas9, natural Cas9 cuts the DNA opposite to each other, so generating a blend-ended DNA molecule. Right? Do you remember that? But the CPF1, on the other hand, it cuts the DNA almost 5 nucleotides before the end of the Proto-Species. Okay? Here at this position, there is one cut, and then 5 nucleotides in, there is another cut on the double-stranded DNA. Okay? And it generates a cohesive end. Cas9 had a PAM site NGG. Okay, the CPF1 has a PAM site triple T V. V means any of the 3 nucleotides, A or C or G. Okay, so this is 3 nucleotides which need to be very specific, and one is variable. So it means that Cas9 has more frequent sites because it requires at least double G, which is specific. This requires triple T. So the chances of occurrence of 3 nucleotides are less than 2 nucleotides. But still, they are much more than 4 nucleotides or 5 nucleotides. The other characteristic feature of CPF1 is that it makes a cut far from the PAM site and generates a 5 nucleotide overhang. So makes a cut on one double-stranded DNA molecule at this position, and then 5 nucleotides away on the second strand. Okay. One of the questions is this site very specific? The answer is generally no. The CPF1 does not cut the specific sequences. It counts the nucleotide and makes a cut whatever these sequences are. But when you are doing genome modification out of a particular nucleotide sequence, you can predict that at what position it will be coming and what would be the protruding overhang. Okay? So you know it because you know the target sequence. And then you can add the same overhangs to the DNA fragment that you want to replace between the 2 CPF1 gRNA complexes. Just like these systems, you can design 2 complexes of gRNA and CPF1 to generate 2 cuts, and then you can delete the DNA in between, okay, and insert the DNA there, which is either repaired or it is having a gene of your interest. So you can use either CPF1 system or the Cas9 system. I was telling you the difference between the two. The Cas9 always generates a blunt end. A CPF1, on the other hand, it generates a protruding end. And you know the sequence of protruding end, and you can design a complementary sequence on the DNA fragment that you want to replace or insert at that particular position. Okay, so this is advantageous for doing DNA repairs or inserting a foreign gene complex here. So usually I have seen people preferring the CPF1 system, okay, when they are doing the fragment insertion, whether for repairing the mutation or inserting the foreign gene. And CPF1 is therefore a CRISPR-associated two-component system. Okay, RNA programmable means you have to design a gRNA, and that gRNA has the protospacer that binds to the target DNA. That's why it's called RNA programmable DNA nuclease. It's directed by the gRNA. Okay. Target DNA is cleaved as a 5 nucleotides staggered in sticky cut distal to the 5' PAM. So it is always away from the PAM site where the cut is placed. Again, you can recognize the site on the target sequence when you are designing the protospacer on there. So this staggered ends at the end of the cuts, they actually support homology dependent repair, HDR. The other one was non-homologous end joining. You just join the blunt ends, okay? And this may introduce mutations. It can cause knockdown of the genes. We will see a similar slide later on during the lecture as well, okay? A very good application of CRISPR-Cas system is just like RNAi. Have you heard about RNAi?
Yes, sir.
Okay. RNAi is used to suppress the gene expression, right? Just like that, you develop the CRISPR-Cas system to suppress. Suppose you have to suppress a gene. How you can do that? How you can suppress a gene? Simply, you occupy the promoter region and don't let the RNA polymerase come and do transcription. If there is no transcription, there is no gene expression. Is it clear? Does it make a sense?
Yes, sir.
Okay, so what they did in this case was they used Cas9. Cas9 is a defective Cas9. It does not have the capability to cut the DNA. Either of the strand cannot be cut. This is Cas9 is incapable of cutting any of the strands of double-stranded DNA. But it has the capability to bind to the gRNA, and gRNA has the capability to bind to a specific sequence of target due to the complementarity of the protospacer. It means that the whole complex is targeted just like the natural way. The enzyme itself, the Cas9, Cas9, and the gRNA complex then go and bind to the target sequence. But The Cas9 does not have the capability to cut. So it will keep on sitting there. If you design this gRNA against sgRNA or gRNA, meaning same thing, okay, single guide or guide RNA. Okay. If you design a gRNA against a promoter region where the RNA polymerase binds, so what will happen? This enzyme will go along with the gRNA and just it will occupy that place. So resulting in the suppression of gene expression. And that can be enhanced if you fuse the dCas9 with KRAB. Okay? KRAB is simply a transcription deactivating protein. Okay? So this is, you make a fusion protein of dCas9 with KRAB protein, which is a transcriptional repressor. Okay? So it more strongly represses the transcription process. So you knock down the gene expression using a fusion product of defective Cas9 and KRAB protein, which is transcriptional repressor. You design a gRNA against a promoter sequence. It will take this dCas9 and KRAB fusion protein to that region and it will stop the gene expression. So it is analogous to RNAi. I would say it is more stronger. It suppresses gene expression more efficiently than the RNAi complex. Okay. So it means CRISPR-Cas system has some other utilities too. Not only it is used for DNA repair and transgene integration, but it can also be used for gene inactivation or gene suppression of gene expression. Okay, that's one thing, and this is depicted in these 4 lines here. Another application of CRISPR-Cas9 system is the activation of gene expression. This CRISPR-A system is actually the further activation of the expression. This activation results into the expression of a gene at a very high rate. So what you do is that you fuse a transcriptional activator like VP16 to the defective Cas9, okay? And then you design the gRNA against a promoter, where the polymerization or the transcription of the gene occurs. And this complex goes there, this transcriptional activator enhances the binding of RNA polymerase II to the sequence or the target of our interest. Okay, so in this way we can actually enhance the expression of a gene by about 25-fold. Okay, so what I was telling you was that CRISPR-Cas9 system or CRISPR-CPF1 system can not only be used for DNA repair, but they can be used for transgene integration and also suppression of the very specific genes if we use CRISPR-I approach, or we can enhance the expression of a gene if we use a CRISPR-A approach utilizing VP16. Okay, is it clear or you need any further explanation of that?
It's clear, sir.
Okay, that's fine. Now coming back to this slide, and this is the one that I showed you early during CRISPR-Cas9. That also in the zinc finger proteins. Okay, I showed you this slide. That whatever this CRISPR-Cas9 system does, it cuts the DNA, and the DNA can be repaired by two systems. One is homology dependent repair. It means you have the terminal ends of the target DNA and the DNA you want to replace it there. They have the same cohesive ends. You can join them together much easily. If you have cohesive ends, you can go for it. It is much easier to do HDR. Okay. If you produce blunt ends or non-cohesive ends, or the ends which are non-complementary to each other, then you go for non-homologous end joining. Okay, and this part, what it does is it produces indels. Indels is insertion for IN, DEL for deletion. So this may result into insertion of a deletion of nucleotides, resulting into a mutation. Okay, and that mutation may be lethal. That may switch off or knock down that particular gene. If you have double-stranded DNA breaks which are close to each other, okay, then you can lose this fragment because these ends are usually blunt. A blunt end can join to C blunt end, and you have a big fragment deletion in that case. Or if you have on two homologous chromosomes, you have the breaking of the double-stranded two similar positions. It can result in translocation of a segment of DNA from one chromosome to another one, resulting into translocation. Non-homologous end joining is never preferable until and unless you really want to mess up the DNA. Okay, you want to disrupt the gene. So multiple double-stranded breaks at one position, another position, and another position due to non-specific binding of the gRNA molecules can cause multiplexed gene targeting. So you get up with many other mutations which are mostly undesirable, and they are very much analogous to the off-target sites in case of CRISPR-Cas system. Homology dependent repair. You can repair the DNA, you can insert a transgene, and insertion of the transgene, if you look down here, this part of CRISPR-Cas use is analogous to transgenic technology. And these transgene insertions, they undergo the biosafety regulations. Okay, while the repair of the gene, you are not inserting anything, and there is nothing which is problematic here.